Hello, this is video number 6, and this is the final video for Module 5, Extensions of Mendelian Genetics. In previous videos, I've been covering extensions of Mendelian genetics based on a single gene which altered the monohybrid cross ratio. In this video, I'm going to be talking about extensions based on traits determined by two genes, but it still involves an alteration in the expected Mendelian ratio. And this is modeled on Mendel's dihybrid cross. In the dihybrid cross that Mendel did, he crossed together strains that differed for two traits, and he found in the F2 generation of a dihybrid cross a 9:3 to 3:1 ratio. So that's the Mendelian ratio. And for all of the extensions that we'll talk about, the dihybrid cross will produce an altered ratio or an altered pattern in that ratio. So why is an altered ratio or altered pattern observed in these extensions? The reason is that the two genes involved are somehow cooperating or interacting to produce a given phenotype. In the example that Mendel looked at, the genes that were involved in P color and P shape were not at all interacting together. They were completely independent. But in these examples, the altered ratio results from genes that interact, and we'll look at examples of how that can occur. Here is the standard cross that we'll be looking at in all of our examples, and this is basically the dihybrid cross that Mendel set up. So he had pure breeding strains, and we'll look at the same example. In the parental generation, individuals that are homozygous for two genes, in this case, homozygous wild type, that's what the plus sign indicates for gene 2. Homozygous mutant for gene 1, crossed to an individual that's the opposite genotype. Wild type for gene 1, mutant for gene 2. In all examples we're looking at, the mutant alleles are null alleles and they produce non-functional products. The wild type alleles produce functional protein, typically enzymes. From the dihybrid cross, the parental generation will produce an F1 in which all individuals are heterozygous at both genes. And if you cross together F1, this could be selfing in plants or mating between different a male and a female in animals, you get the F2 generation. Remember, Mendel saw a 9:3 to 3:1 ratio. And all of the examples we'll look at today, that ratio is either altered in number or in some other way. If genes do not interact, this is what Mendel saw, we would expect to see the Mendelian 9:3 to 3:1 ratio in the F2. If they do interact, this ratio is going to be altered, usually by combining one of these classes or two of these classes together. For example, one alteration is 9:3 to 4, and that comes about by combining these two classes, the 3 and the 1. Just to remind you, this is the result that Mendel saw in a dihybrid cross, crossing together pure breeding pea plants for pea color and pea shape, yellow and round, crossed to green and wrinkled, gives an F1 in which all individuals are heterozygous for both genes, displaying the dominant phenotype yellow and round. And then a cross between F1 produces in the F2 the 9:3 to 3:1 ratio. Okay? So this is what the basis is, and now we're looking at examples that alter from this somehow, and these are why it's considered an extension of Mendelian genetics. So these are the examples, and this is straight from the outline. And we're going to look at examples, at least one example for all of these 5 listed. And there may be more examples in the PowerPoint, but for the sake of brevity of the video, I'm just going to choose among them. First example of an extension is called Gene Interactions, producing a novel phenotype. And this is seen, the example I'm going to talk about is seed color in lentils. This example, milk snake color, is shown in the PowerPoint. All right, so here's the cross, dihybrid cross, seed color in lentils. We're looking at the shape of, the color that you see in different plants with different genotypes. So here is pure breeding plant producing tan seeds, genotype of pure breeding plant producing gray seeds. If you cross them together, you'll get an F1 that's heterozygous for both genes. Notice that the phenotype of the F1, unlike what Mendel saw, the phenotype of the F1 doesn't resemble either of the parent phenotypes. Okay, so that's a novel phenotype. And if you mate two F1, you do produce the expected numerical ratio, 9:3 to 3:1. However, amongst those are phenotypes that are not seen in the parental generation and even in the F1. So 9/16 having both dominant alleles produces the brown phenotype. So that's what we saw in the F1. Having only dominant for A produces the tan, only dominant for B produces the gray. Being homozygous recessive for both pops up even a new phenotype, green. All right, this is a departure from Mendelian expectations because these new phenotypes crop up brown and green. Why is that? That's because there's interaction between these two genes in producing the phenotype. And the interaction is this. Gene A encodes an enzyme that's responsible for producing the tan pigment. Gene B produces an enzyme that produces the gray pigment. If both of these enzymes are present in formation of the seed, they combine to form the brown phenotype. If enzyme A is missing because it's homozygous for the null A allele, only the gray pigment is produced, gray phenotype. If enzyme B is missing, homozygous recessive for B, only the tan pigment is produced, tan phenotype. If both enzymes are missing, neither tan nor gray is produced, and the phenotype is green because now chlorophyll, deeper within the seed, is the pigment that's observed. All right, so this is a departure from Mendelian, not in numerical ratios, but because a new phenotype pops up. All right, second example is called Complementary Gene Interaction. And we're going to look at sweet peas and the flower color they produce to illustrate this. In complementary gene interaction, a 9:7 ratio is seen in the F2. And that is an indication that these genes are somehow interacting in production of this flower color phenotype. All right, here's the metabolic pathway in blue-eyed Mary plants that's involved. There's a colorless precursor, which is converted by an enzyme encoded by the gene of the C gene. And if that enzyme is present, it converts it to a colorless intermediate. The colorless intermediate is converted by an enzyme encoded by the P gene. And if that enzyme is present, it produces the end product for this pathway, which is anthocyanin, a purple pigment. Plants that have anthocyanin are purple, produce purple flowers. Plants lacking anthocyanin produce white flowers. So here's the dihybrid cross. In the parental generation, pure breeding strains, homozygous for the wild type allele for C and the null allele for P crossed to the opposite genotype produces an F1, heterozygous for both genes. Notice that this plant has purple flowers. It has a dominant allele for both, meaning it can produce functional enzyme C and enzyme P and produce the purple pigment. When you do an F1 cross, doubly heterozygous for both genes, you produce this Punnett square, and which is representing, for a Mendelian ratio, 9:3 to 3:1. However, the phenotype that's observed is this: 9/16 showing purple flowers, and the 9/16 with purple flowers have at least one dominant allele for both C and P genes. The rest, 7/16, are homozygous recessive, containing null alleles, for either the P gene or the C gene, or both, because they lack functional enzyme P or C or both. They cannot proceed all the way through this pathway to produce the purple pigment. So in this type of Mendelian extension, these genes are interacting in a common metabolic pathway, and the altered ratio is a result of that. All right, next example is what's called epistasis. First, a general definition of epistasis. Epistasis is when the expression of one gene masks the expression of another. So this is basically a dominance-recessive relationship between genes. Unlike what we've talked about so far, dominance is between alleles of a single gene. In this case, the genotype of one gene is masking the expression of another gene. Epistasis also indicates that the two genes are somehow involved, interacting in the production of a particular phenotype. There are two types of epistasis. Recessive epistasis is when a homozygous recessive genotype at one gene blocks the expression of wild-type alleles at a second gene. Dominant epistasis is when a dominant allele of one gene blocks the expression of wild-type alleles at the second gene. Let's look at examples of both of those. And this is just to remind you that epistasis is indicating interaction between the genes in some common pathway. So first example we'll look at is recessive epistasis, and I actually have two examples I'll talk about: flower color in blue-eyed Mary plants and coat color in Labrador retrievers. In recessive epistasis, the F2 ratio in a dihybrid cross is not 9:3 to 3:1. It's 9:3 to 4. And let's look at that altered ratio in blue-eyed Mary. All right, so blue-eyed Mary can have blue or magenta or white flowers. And the flower color is determined by two genes, the W gene and the M gene. The W gene produces an enzyme that converts a colorless precursor to a magenta intermediate. The M gene produces an enzyme, enzyme 2, that converts the magenta intermediate to the blue end product. If you have functional enzyme 1 and 2, blue flowers result. If you have only functional enzyme 1, magenta flowers result. If you lack enzyme 1, regardless of whether you have functional enzyme 2, the pathway is blocked at this step and white flowers result. Okay, so in a dihybrid cross, you would see a 9:3 to 3:1 ratio in terms of genotype, but the phenotypic ratio is 9:3:4. And that's because being mutant at the W gene results in a lack of production of enzyme 1 and no formation of the magenta intermediate. So this is recessive epistasis because being homozygous recessive at the W gene blocks expression of the wild-type allele of the M gene. Second example of recessive epistasis is in Labrador retrievers. And in Labrador retrievers, there are 3 varieties: black labs, chocolate labs, and yellow labs. There are 2 genes involved, gene B and gene E. In this example of recessive epistasis, being homozygous recessive at the E gene, is going to block expression of the B gene. This shows how that works. It'll be in a dihybrid cross in dogs. If you cross a black lab, which is homozygous dominant for both genes, to a yellow lab, which is homozygous recessive for both genes, you will get an F1, which is heterozygous for both genes. These are black labs, resembling the homozygous dominant parent. If you cross these two black labs, F1 black labs, together, then if you have a large enough population of dogs, you would expect to see the Mendelian ratio of 9 to 3 to 3 to 1 based on the genotypes. However, what you observe is instead the 9 to 3 to 4 ratio characteristic of recessive epistasis. The 9/16 have a dominant allele at both genes, 3/16 recessive for B, dominant allele at E. 4/16 are combinations as long as you're homozygous recessive for E, regardless of the B gene genotype. Then you are the yellow lab. Why is that? This slide explains the process. So the E gene and the B gene are cooperating in this phenotype. The E gene produces an enzyme, protein E here, which converts a colorless precursor to the dark pigment eumelanin. The B gene produces a protein B. This is not an enzyme, but this protein is involved in depositing this pigment in the fur of the animal. So if you have functional protein E and B, you produce the dark pigment and you deposit a lot of it densely in the fur. Dense deposition results in the black lab phenotype. If you have functional protein E but lack functional protein B, you still produce the dark pigment, but you're not able to deposit it as densely, and the resulting phenotype is the brown or chocolate lab. If you're lacking protein E, regardless of whether you're producing functional protein B or not, you don't make the pigment. So without the pigment, you can't deposit it. And the colorless precursor is shunted into this alternate pathway, which produces an alternate pigment, a lighter pigment than eumelanin called pheomelanin, and that gets deposited and produces the yellow lab phenotype. So there's a 9 to 3 to 4 ratio due to the fact that the genes here are producing proteins, cooperating and producing these different phenotypes. I have one example of dominant epistasis, and in dominant epistasis, the presence of a dominant allele of one gene masks the expression of another gene. And this is seen in summer squash. If there are two genes, the A gene and the B gene. If you have a dominant allele at each, you produce white squash. If you're homozygous recessive each, you're green. If you cross two pure breeding white and green together, you get squash plants that are heterozygous at both genes. And they produce the white phenotype, same as the parent here. If you cross two F1 squash plants together, you expect to see a 9 to 3 to 3 to 1 ratio in terms of genotype, but the phenotypic ratio observed is 12 to 3 to 1. And the 12 is a combination of the 9 and 3 genotypes. Why is that? This slide explains the function of the proteins encoded by the A and the B gene. So in squash, there's a green pigment that can be converted by enzyme A. If squash have functional enzyme A, it's converted to yellow pigment. Protein B is involved in blocking the deposition of this pigment in the squash. So if you have functional protein B, whatever pigment is made is not deposited, and so the squash is white. Now, if you lack enzyme A, this catalytic pathway is blocked. And no yellow pigment is produced, but green pigment is still present. However, same as before, if protein B is present, that's going to block deposit of this green pigment, and the squash is white. If you have functional enzyme A but no protein B, yellow pigment will be made and deposited because no protein B there is present to block. And yellow squash is the result. If you lack both enzyme A and protein B, the green pigment is present and it will be deposited, and the green squash results. So the altered ratio is due to the interaction of these two genes and their protein products and this particular phenotype. There is an example of dominant epistasis, which produces a different ratio in chicken pigmentation. That's described in the PowerPoint if you're interested. The last example of genes interacting in production of a phenotype that alters the Mendelian ratio is seen for redundant genes in corn or maize. And this slide shows in maize there are two genes, the A gene and the B gene. And as long as you have a dominant allele of either A or B, they're basically providing the same function. If you have a functional allele of A or B, you'll produce this phenotype, the broad leaf. If you lack functional allele, homozygous null for both A and B, this produces this skinny leaf phenotype. So if you do a dihybrid cross between these two genes, big A, little b, cross to little a, big B, you produces an F1 that's heterozygous for both, and then cross two F1s together. This is the Punnett square for the 16 genotypes that result. And the ratio here, 9:3 to 3:1 in terms of genotypic ratio. But it's a 15:1 phenotypic ratio. 15/16 have at least one dominant allele at one gene or both. And they are going to produce the wide leaf phenotype. 1/16 only will be homozygous recessive, having only null alleles at both the A and B gene. And this produces the skinny phenotype. These genes are redundant in function. They both provide the same function, so you only lack that function being homozygous recessive for null alleles. And this is showing the idea, as long as you have a functional A or B, and that will happen for these first three genotypes, you're going to be able to recruit leaf cell precursors during development of the maize plant and produce these broad leaves. If you lack both protein A and B, that recruitment is not occurring, and the leaf is very skinny as a result. So this slide is a table summarizing the examples that we went through, and it gives you the example and the phenotypic ratios that were seen, and how that departs from Mendelian ratios. It's a good summary table. So this ends material for Module 5.